So what are you guys doing for Guns N' Roses?
Yeah, don't ask me to name a song though. I hate when people do that. I'm just like, it's just a shirt. Like, please let me wear it in peace.
I apologize for breaching your peace. Any plans for the weekend? I know. Okay, so on the subject of sleep, my day started at 1 a.m. and it hasn't stopped yet. So I'm 11 and a half hours into my day. So if I stare blankly for like 60 seconds, just make the most of that time. Do some home. If I just stop and just stare into some abyss somewhere, do some homework, send an email. I'll pick back up when I wake up again. I'm gonna have some micro sleeps during class. So I think JP, I'm on your sleep schedule for today. So we will wrap up all of the just genetic for genetics sake stuff today. And then next lecture is going to be exercise genomics, more application, employment of genetics, and age. What happens to us with age, looking at things like telomeres and how exercise is an intervention from a genetic expression and telomere perspective. So there's a little bit more application, a little bit more context in the next lecture. And I think there's going to be a guest lecture next week. Travis Stiles, some of you have met him or heard of him. He is the president of Novaron Bioscience, and he did his PhD in neuroscience at UC San Diego. We did our undergrads and master's degrees together, and then we got about as far apart as you can. I was in Connecticut, and he was in Southern California for our PhDs. And he talks very fast. So it'll be recorded, and I think what we're gonna do is do a Zoom. We're gonna do a Zoom call, and probably just put them up on the screen, and in a way that we could engage. But it'll be recorded, so if you need to like put it back on slow-mo, sometimes those words come out like little racehorses. So wrapping up the genetic stuff for today, just always remember this, just keep this stuff as thought one. When you encounter. Thought zero can be charm. Thought one has to be, let's grade these people. We're gonna get back into just one day of this and a competition of that. We'll talk about it from the perspective of metabolism and application in the same day, the meaning as well as the breaking down the biochemistry of it. Big picture mTOR stuff, don't forget that. Now, remembering that all of those P experiments that we had, Gregor Mendel, those P experiments, this didn't put genetics on the map or inheritance on the map. I'm not going to ask for this section, I'm not going to ask questions specifically about people's names, but for context's sake, Gregor Mendel, this is late 19th century, and yet, you can look at the Old Testament, Talmudic law, and we're looking at inheritance being identified, that if a woman had 2 or 3 sons who died from circumcision, bleeding, right? Not from like the terror of it or whatever, but like from the bleeding, subsequent boys should not be circumcised under the assumption that they too will die, that there is an inherited quality. So this is thousands of years ago. And even in families, it's not just the individual, looking at lineages, looking at families. Yeah, don't write this down. This is just context that we've known about inheritance for an awfully long time. It once happened with 4 sisters from whatever that place is. That the first had her son circumcised and he died. The second sister had her son circumcised and he died. The third sister had her son circumcised and he also died. And the 4th sister came before the rabbi and he told her, you must not circumcise your son. Inheritance. Inheritance has been understood to some degree, to some practical degree for a long time. Now, the date palm is probably the first plant. There's enough literature that talks about the first plant being hybridized, and let's get some other pollens in here, and let me take some of that ancestry and mix it with this ancestry. But plant hybridization is super old, only thousands of years for this history. And animal husbandry, breeding, selective breeding of animals, horses, thousands of years, certainly in the last few hundred years, it's become, really bottleneck, but it's become this high dollar field of selective breeding of horses. But tracking footprints of artificial selection in the dog genome, the size, shape, and behavior of the modern domesticated dog has been sculpted by artificial selection for at least 14,000 years. Artificial selection, let's breed the ones with floppy ears that smile and lie in my lap and bark at the enemy. Whatever traits, selective breeding of traits. We've known this for a long time. He's the father. Mendel is the father of genetics, but concepts, ideas of what inheritance is and how inheritance passes, this is not a new line of thinking. I mean, this is thousands of years old understanding of inheritance. But breaking it down a little bit more of sort of the mathematics of it, being able to make predictions about inheritance, and then certainly looking at alleles, which we'll talk about today. But genes, if you have a genome that predisposes you to something good, something bad, whatever, it increases your odds of fulfillment of some destiny, but it doesn't place you there, it doesn't deliver you there. It depends on the gene's expression, and the expression of genes, remember, depends on context, depends on environment, depends on the application of stresses and loads. So if you are really emotionally stressed all the time, that has an influence. If you undereat or overeat, that has an influence. Even just break down your macros, right? Look at your carbs and fats and proteins. And what is that composition? What does your diet look like? That too influences genetic expression. All of this stuff is altering the behavior of your genes. So you have a genome, you have DNA, which is pretty much steady, and then its expression depends on environmental context. But we're set up with either good responses, bad responses, no responses, it can be negative or positive or non-responders to whatever the stresses, whatever the stimuli are. And some people can just withstand, I mean, what would you say makes an athlete really good and talented and likely to go pro? I have my answer, but I want to hear yours. Genetics, yes. Okay.
I don't know, work ethic.
Work ethic, I would say yes. Very much work ethic, very much, and genetics as an umbrella term to capture work ethic, to capture height and muscle fiber composition, stuff like that. Genetics as a all-inclusive everything. I mean, it's like what makes an athlete good?
Tall, nobody cares if you're good at basketball.
Say that again?
You're 8 feet tall, nobody cares if you're good at
basketball. Manute Bol. Yeah, who's on the first slide? It was Manute Bol and Muggsy Bogues. Muggsy Bogues is like a foot tall, and Manute Bol was easily, the tallest building in Chicago. He wasn't in Chicago, but genetics is going to have a huge. But what else? What other things are going to determine likelihood of athleticism or signing a pro contract or whatever?
Income, how you're raised.
Oh yes, very much so. Yeah, the income. There's at the end of this, I've started putting together the next article, and I have one. I think it's from 2021 about income and the likelihood of success in athletics. And if you come from a wealthier family, your likelihood of success. People have this fairy tale story about, basketball was my escape. That's what got me out of my neighborhood or whatever. Or it was football, it could be baseball, it could be any sport. That was my escape. And that's more of a fairy tale than, certainly than Robin Hood, which is, I don't know, has a weird history. But one thing that's really important though, is the ability to withstand injuries. And there is a genetic relationship with vulnerability or durability, vulnerability to injuries or durability, the ability to withstand. You send somebody into training, and most people have if you talk to people who are good at sports, they were good and they have a story about why they didn't go pro. It's, oh, my knee, or it's my shoulder, or my ankle, or my whatever it is. Yeah, but that's genetic too. And that's the type of genome that would have never made it because of an ankle or a shoulder or a knee or whatever. That too is part of the whole package. And that package is not determined by, but heavily, heavily, heavily influenced by genetics. So again, we have predispositions to things. People who can withstand, just the emotional onslaught. There are people who can work a 100-hour work week and still get to the gym and then smile at people. There's that class of rare creatures. Other people, it's a 5-hour work week and they're like, you know what, I quit. And so, but there's a lot of genetics here. There's conditioning, there's psychology, there's conditioning, but genetics play a huge part. Tons of functions of protein. Do remember that part, a bunch of different functions of protein. At the end of today, I'll have a slide with a bunch of, here's the stuff I want you to remember about genetics as we move forward. With applications of load, with stresses, we change our protein values, but we don't all change the same. We don't all express those proteins in the same way. Now, again, I'm not going to ask about names, Watson and Crick, and Maurice Wilkins won. That was the Nobel Prize, but that was just deciphering the chemical structure. And again, the only chemist in that group was Rosalind Franklin, and who died at 37 or something of ovarian cancer from all of those X-rays. One can make a rational leap that it was from all of those X-rays as opposed to just, it happened. And but this was a critical step in understanding the nature of genetics, the chemistry of genetics, but the applications of genetics came later. We had to have that foundation first before we could make any real progress in understanding how genes beget risk or set us up, position us for success. If we're going to do a talent identification system, let's look at candidate genes and see what sports I'm most qualified for or most likely to succeed in. That is still young and weak. There are some candidate genes that have emerged strongish, but remember, that's the gene that makes me a basketball player. That's the gene that makes me a tennis player. That's the gene that makes me a distant. Constellations of genes contribute. It's not like there's one bright star in the sky and that's the thing that determines whether someone excels at something. There is a constellation of alleles, a constellation of genes that can contribute. So remember, a genome, that's the collection of genes. Everyone in this room has a unique genome. Unique genome belongs to everyone in this room. And a gene is just a sequence of DNA. And the protein coding genes, we encode proteins. It's a recipe to link together amino acids in a very specific way to create a protein. So a gene is a sequence of that DNA that encodes a protein. And then chromosomes, we have 23 pairs of these things, autosomes or allosomes for the sex pair. You don't need to know that. Just you have 23 pairs of chromosomes, 1 through 22, right? And then we have XY or XX. And that's where all of our DNA lives. That's where our genes live, is in these chromosomes. Now, again, if chromosomes are books and genes are the chapters, each book, it varies widely, but each book has roughly 1,000 chapters. If you do the mean value, each book has about 1,000 chapters. Now, each chromosome has about 100 million base pairs. 100 million, and again, varying. T goes to A, C goes to G, G goes to C, A goes to T. If you're an RNA, you replace that T with a U. And those are the base pairs, right? It's easy to remember what the base pairs are if you're a musician. because C major, G is the perfect fifth of C major. There's songs, if you look up, who's a musician? Like, who plays instruments? Oh yeah, Kevin, definitely. Sometimes you have your instrument here. There's a lot of songs that are just G and C. Paperback Writer by the Beatles, nothing but G and C, right? So G and C, I think Mary Had a Little Lamb, I think that one's just G and C. But just go back and forth, G and C, it sounds nice. And so G and C go together. I know Kevin's gonna remember that, and I know some other people will. For everyone else, maybe TA for like teaching assistant, or AT for athletic training, or what, you know, I don't know, whatever. But T always goes with A goes with T, C with G, G with C. Now, when you get into RNA, you swap out those T's for U's for uracil. So this would be what, U, A, C. In terms of the translation, what we're going to be translating, I think that's tyrosine, look it up, I don't know. So, about 3.2 billion base pairs. Okay, 3.2 billion base pairs. Now, not all of that. It used to be, a lot of this used to be called, don't write this down, junk DNA, meaning it's not coding for a protein, so it's not doing anything. And that's not true. There are regulatory functions for a lot of this that's not directly coding for a protein. But each one of those nucleotide triplets, they work in threes, right? They couldn't work, certainly you can't work in ones because there's 20 amino acids. You have to have a recipe for 20 different amino acids. Can't work in ones. If there's two, not enough to code for 20 different proteins. If there's three, okay, we're a little bit over, but a little bit over, it's perfect. Because there's four, we're like way over. So three, there's something elegant, that's the perfect amount, is a triplet, a nucleotide triplet. But we don't express proteins everywhere ubiquitously, right? So think there's hair over here, and there's a pancreas over here. Beta cells in the pancreas, what do those spit out? Insulin. Are you spitting out insulin in like these cells? Or are you like growing hair? Growing hair, right? Are you growing hair in your pancreas?
You shouldn't.
You shouldn't be. If you are, happy Halloween. I don't know what else to say. So depending on where you are in the body, you are expressing some genes. If you're somewhere else in the body, you're expressing other genes. You're not expressing every gene everywhere all the time. Right, so this is a nice little summary of that, which is 23 pairs of chromosomes, counting the XY, right, 1 through 22 and XY, contains approximately, 20-something thousand, 23,500, who cares? 20-something thousand protein coding genes, protein coding segments known as exons, and then there are these intervening sequences, these adjacent sequences are the introns, and that's what people used to think of as this like junk DNA, because it's not directly coding these proteins, but there are regulatory functions. We're not really going to talk about them, but down here, approximately 99%, approximately being one of the key words, approximately 99% of the human genome does not code for a protein. So when you look at all of these base pairs, man, there's a lot of them. Oh, 99% of the human genome is not coding for a protein. However, these regions, by and large, which is a stupid term, that's a nautical term that means like with and against the wind. Actually, I dislike the grammar in this because comprise means contain and is contained of 3 point, that doesn't make sense. It does it again, comprise. So I dislike the grammar, but the paragraph is nice. Content, science, that part is nice. 99% of the human genome does not code for a protein. However, these regions, by and large, have biological functions that might affect gene expression and likely the clinical phenotypes. So we have, between a chicken and a grape, right, low 20,000s, protein coding genes. And then there's a bunch of base pairs that have other functions too. But each one of us has a different number of protein coding genes. We have a different number of genes, right? It's not like the human genome from the perspective of we all, we're 99% the same, but that 1%, man, that means a lot. That little difference means an awful lot. And getting back to this idea of the number of genes somehow mattering, when people would talk about like, well, we're not even twice, but we're nearly twice what a fruit fly has. Okay, but it doesn't matter. It matters what the proteins are and what the proteins are doing. Having a ton of base pairs, what good does that do anyone? What we need is the right proteins serving the right roles. And so to get those proteins, there are some additional ways. Let's say we have 23,000, something like that, protein coding genes. That doesn't mean there's just 23,000 proteins, right? So there's alternative splicing. One gene can make different variants, can make different versions, different isoforms, different versions of proteins. So you can get different proteins from a single gene, and post-translational modifications. You can modify those proteins once made to serve different roles, to take on a slightly different role, slightly different functions. So that lands us, and also, we have a bunch of stuff that we're just not expressing. Atavism. Remember, we have these relics, these genomic relics that we're not expressing. So how many proteins do we get? I don't know. We'll figure out that question at some point. We'll have an answer to that that is meaningful at some point, but we're nowhere near that now. But we have a lot of proteins, and we don't seem to have that much useless stuff. What we first identified is, just mostly useless stuff, and there's a little bit of functional stuff inside of us. That's not quite the story. We just haven't figured out what everything does yet. And even the proteins that we know quite a bit about. Take something like nebulin, or let's get into the muscle. And is that serving mechanosensation roles? Yeah, probably, but has anybody really outlined that? I've never seen it. And so there's a lot of the proteins that we have a good understanding of them. We don't have a complete understanding of them. And those are the ones we know most about. And there's proteins that we don't know about. There are proteins we've identified, and we don't understand their functions. So proteins, these are doing everything in your body, and we know remarkably little about it. But this is where they come from, right? Genetic expression. This is where those proteins are emerging from. And you're half your mom, half your dad. There's you. Again, we have these base pairs. And so this is DNA, not RNA. And you know that because you see a T, so it has to be DNA up there. These are our nucleotides. We need 4 of them, like digital, like zeros and ones. You can create a pretty impressive whatever in a bunch of zeros and ones. But to create our genetic alphabet, having 4 letters really is ideal. Again, we have our symphony of life with 4 notes. We have our sort of the great story of biological history, human history, squirrel history, dendrology with trees and whatever. Life, biology, the story of life is told with a 4-letter alphabet. 5 keys, A, C, G, T, or U, right? So these ones, it depends if you're DNA or RNA. DNA, nucleus, right? It is in the nucleus. You transcribe it. That can take a while. Transcription can take a while. Get your RNA, messenger RNA, and take it to the ribosome and translate a protein, link together a bunch of amino acids to create a protein. There's also transfer RNA. I don't know if you see it in any of these. I don't think it's any of these, but so you have to actually get the amino acids there. The amino acids don't have minds of their own. Like, it's work time. Amino acids don't do that, so you have to actually get them there. But the mRNA, so when you see that, the little m, messenger RNA, that is the recipe that you're going to take to the ribosome to read and interpret and link together all of those amino acids to create your protein. Again, we have a large subunit and the small subunit, and the codons are what are being read. Those are those nucleotide triplets. Each one of those is coding for a very specific, I mean, it look like little gumballs in this diagram, but each one of those codons, you'll have a start codon, a stop codon, and then everything in between is some specific amino acid. All right, put this one down, put that one down, put that one down. So genotype, that's your genes, right? That is, I mean, you can figure out, you can do you can sequence your entire genome and figure it out when you're an infant, right? Before your phenotype has really presented itself, before you've exhibited any phenotypic traits. You're just like a little, oh, to me, like every fetus looks the same. Like boys, girls, every race, everyone. I can't tell the difference between one fetus and the next. So phenotypic traits, these kind of take a little while. These take a little while to develop. So we have our genes, and then we have our phenotype, which this is the proteins make this. What proteins you express make your phenotype. And then with our hands and our tools and whatever, the extended phenotype is what we make with the proteins we were given, the resources we were given. Remember, bees make hives, birds make nests. I mean, just animals do their specific thing. And us among them, we do our specific thing, but we make like minivans and castles and stuff. So how do you make your genes express themselves, do stuff, anything you do, any load, any stress, anything, environmental or applied. You know, again, I was like up since 1 a.m. My genes are being expressed differently than if I had slept like a normal person. My genetic expression profile has been changed. Anything you do or don't do changes genetic expression. And when we see translation down here, with mTOR over here, hypertrophy, protein translation, hypertrophy, this, we know how to turn on or turn off translation in this sense. So again, a codon, triplet of nucleotides, deoxyribose, that's the, when you see the sugar, the 5-carbon sugar, so this is DNA because you see the T, so that's deoxyribose. There's ribose is the RNA. You know, who cares? You don't need to write down. There's the difference. Almost nothing. But the RNA is your recipe, right? The RNA is your recipe, and you see those U's. You see U's in the place of where the T's were. U's in the place of the T's. And UAG, right? That's going to be our stop codon right there. And then the start codon, the AUG, methionine. Let's start with methionine, and then we'll code for very specific things. Individual amino acids, hit the stop codon, and let's get it out of here, right? We have assembled a really tiny protein. And so each one of these has, again, leucine, there's a bunch. There's a bunch for leucine. UUA, UUG, CUU, CUC, CU. There's a bunch of these for leucine. But to get the 20, you need 3. There's going to be some overlap. There's going to be some overlap with these things. But there's stop codons, UAA, UAG, UGA. Those are all stop codons, meaning you hit that last one and the process is done. You have crossed the finish line. Your protein can leave the nest. And then the start codon, methionine for the start codon. So the process always begins with a start codon, and then you have these intermediates that each link an amino acid onto that chain. And then you have a stop codon, which halts the process. You are done. So the codons are important because this is the sequence of amino acids, and the sequence of amino acids determines what the protein is. The protein that you get is determined by the exact sequence of amino acids. And we know what these proteins are, right? A throwback to like just now for these slides. The hormones, the binding proteins, all of our enzymes, I mean, everything that makes your muscles move, everything for digestion, pumping around ions, all of these verbs that your body is doing with or without your conscious thought. All of this stuff is doing, it's a bunch of proteins doing it. And we don't have the same, I mean, this is like Mary-Kate and Ashley, is that their another name? Full House when they were little children. And then the Dursleys neighborhood from so dreaming of identical houses. Now, if you do, like Brookside, just go out to March Lane and go out in Brookside. My first year when I was a student here, to 05 or something like that, I lived in a house in Brookside, and I had to look at the address number on my house because I was at risk of trying to open the wrong house's door. Every house I was in, Nostalgia 3 or 2 or 4, or one of the Nostalgias. I think there's only 3, but it goes up to 4. One of the numbers is missing. But every single house is the exact same. But like if you scrutinize the tree in the yard, it's lemon. Oh, I think mine is lime. Like those are the differences. Once in a while, one is mirrored, something like that. The shutters are different, or the shutters or whatever. And so there's little tiny differences between these houses in the Dursleys neighborhood, Harry Potter Dursleys neighborhood. Now there's no difference here other than the environmental factors and expression, but they are identical, aren't they? This isn't like, okay. But with genes, we have alleles. We have different versions of the same gene. It's like those cookie cutter, whatever, neighborhoods where every house is the same, but this one's mirrored, but this one has the garage door looks different, but this one has the lime tree. There's a little difference in these things, but a little difference can be a big deal. These are called SNPs, or single nucleotide polymorphisms, SNP, SNPs. And this is how we get just a little difference. Right here, you see C obviously goes with G, right? It goes with the perfect fit.
Can you repeat this?
Yeah, SNPs, just as though it's like scissors snipping something, but SNP, single nucleotide polymorphism. And so this is a change. This is a little change in the gene, and you're going to get a different version of that gene. So right here, C to G, that C is replaced with a T. So T goes with A. So we have a SNP. We have a single nucleotide polymorphism. We have a different version of a gene. Now, that triplet, that nucleotide triplet, codes for something else. What used to go there, what amino acid used to be inserted at that particular slot has been replaced with a different amino acid. And those amino acids determine the folding. They determine the shape of the protein. The folding, the shape of the protein is determined by the amino acid sequence. So it folds differently. One different amino acid makes the protein fold differently. Behaviors of those proteins, functions of those proteins depend on their shape. So this, although it seems like a tiny thing, it can be a big deal. You can have a 500, you can have 500 codons, and one of them codes for something else, and the whole protein can behave differently. So it can be important. It's not that it's always important, but it can be very important. Those alleles, different versions of genes, those alleles, different houses in the Dursleys neighborhood, those alleles whatever that you have your little snip here, you have an allele. That's the reason we're different. That's the reason that our genomes are different. So we have these different versions, and maybe we have different numbers of genes, but we have these different versions of those genes. So there's a nonsense SNP. We're going to go over a few versions of a few iterations of genes, of how these things get mutated. And the nonsense SNP is a stop codon, shows up early. A stop codon shows up prematurely. You're not done making a whole protein. If let's say we're taking a test and everyone's writing because the test is super long, right? Because I do that. The test is super long and you're still writing and I say, time's up, pencils down. But I didn't finish the test. Pencils down, that's a stop codon. That's a stop codon saying, no, you are done, but I'm not done. No, you are done, but my protein sucks. I don't care. Stop codon, premature stop codon. What you want to do is get all the way to the end of that linkage, get all the way to that end of that chain of amino acids before you get the stop codon. But if a stop codon shows up prematurely, that's a nonsense SNP. Can be pretty devastating if you're not actually getting the whole protein. A missense SNP is you get a wrong amino acid somewhere in that chain, a missense SNP. You don't stop the synthesis. There's no premature stop codon, but you swap out one amino acid for another. And even if, again, it could be 500 proteins long, this thing, but you get one amino acid different, and the folding happens differently. If the folding happens differently, the behavior functions differently. So, you're making chicken cacciatore, but you put pork in it. That's not chicken cacciatore, it's pork cacciatore, if that's it. I don't even know what this is. I was once making, it was like a vegetable stir fry thing, whatever. And I thought I grabbed cumin and I put a lot of cinnamon on those vegetables. That is not the seasoning that was most appropriate for that dish. It was eaten, but not happily. So that would be a missense SNP, where there's a lot of ingredients. There's a lot of ingredients. Every single one of those ingredients was right, but I swapped out cumin with cinnamon on accident. That was my culinary mutation, and the whole dish was different. Nobody would have taken a bite of it. This tastes like a normal thing that I would expect to be in my mouth. Nobody would have said that because there was one ingredient that was different. Same thing with some of these amino acids, these intermediate amino acids. Now, a frameshift mutation, these can get pretty huge. A frameshift mutation can have a pretty huge effect because if you insert or you delete any of these, let's delete the A. Okay, so what used to be threonine, right? It's not threonine anymore. And what used to be glutamate was those three. It's not going to be glutamate anymore. What used to be leucine, those three, is not going to be leucine anymore. And I can keep going, right? Because what happens is you shift everything. Instead of doing those three, now it's these three. Instead of doing these three, now it's those three. Everything that follows shifts. Everything that follows shifts. And you might find yourself at a premature stop codon that way, depending on the sequence. But a frameshift mutation, that's not a missense SNP, where it's like, well, everything's the exact same, but this one slot. It's like, at this one slot, it changed, and every single amino acid after that is different. It's an unrecognizable protein.
Would you say that's like delete one?
You can delete or you can insert. Yeah. Yeah. So, and you can, we'll talk about some specific conditions at the end that nobody needs to remember any of the specific conditions. That's just a way of illustrating. Let's look at real life examples of what we just talked about. But there are a lot of deletions that are huge. But if you delete an entire thing, you just lose threonine, and then you just move on to glutamate, right? But if you insert or delete one or two, that's a frameshift mutation, and every single codon that follows is different. So it can be pretty drastic. So again, you can just look at what the codes for these things are. And so if we were to delete that one, CGG is, if you find it before me, what is it?
Arginine.
Arginine. Nice, you found it? GP. Arginine. So instead of threonine, it's arginine. It's okay, so I guess over there. So that, let's just put this one all over here, and I mean, everything gets messed up. Everything gets messed up when you do that. So frameshift mutation can be a pretty big deal. Now, why does it matter if we have these mutations? Missense, nonsense, and these frameshift mutations. These differences in our genomes, again, it's not destiny, but it's a heavy predisposition. And it can predispose us to, I'm sure Ronnie Coleman was more responsive in the weight room than the guy lifting next to him, right? There are genetic predispositions. Fat Albert, and that's nobody who I've ever heard of. Is this a famous, does anybody like recognize his face? Like, no, that's a famous marathon runner. So I just took a picture from somewhere. So again, we have Hagrid. He's huge, at least in the movie, not the actor. Or this person who's just chronically tired all the time and has laser visions. Depending on these SNPs, depending on our genome, depending on what genes we have and which ones we're expressing, we take on an entirely different form. I mean, just look around you. We're all relatively different in a number of features. Now, sometimes it manifests in less than ideal ways. Sometimes it manifests in pathological ways. Commonly, it manifests in pathological ways. Now, I would say this particular pathological manifestation is a exceedingly rare one, but there are very common manifestations. So, lecture 11, right? That was our protein catabolism lecture. And we talked about phenylketonuria and taking phenylalanine, you can't see, and converting it to tyrosine. The slides are so blurry and tiny, but to convert phenylalanine into tyrosine, there's an enzyme that will do that conversion, and phenylketonuria means you get this crappy version of that enzyme. You don't do the conversion, phenylalanine builds up. Phenylalanine over-occupies the large neutral amino acid transporters. You can't get the amino acids, the competing amino acids, into your brain, and it causes brain damage, right? So if we have phenylalanine hydroxylase, the enzyme that does that initial conversion of phenylalanine to tyrosine, if we have a garbage version of that, we can't do that conversion. And that's why you see those equal packets talking about this, phenylketonurics beware, it says. Something similar to that as their warning label. This has a bunch of phenylalanine in it. Phenylketonuria, so there's a ton of mutations. And again, don't write down any of the specific mutations at this point. We're just going to go over examples of how genes get mutated. But phenylketonuria, there is, arginine is what you're supposed to have in the 408 position. I think there are 451 amino acids here. And you can translate this thing in probably a minute. But if you, at the 408 position, you do this little crib swap. So you get tryptophan instead of arginine. Now you can't eat an equal packet without sustaining brain damage. One amino acid, right? 450, I mean, there's almost 500 of these amino acids. And you swap out one of them, and an equal packet causes brain damage. One amino acid. Now there's a ton of these mutations. That's the most common one. And it's not that this is a common condition, but that's the most common presentation of it. Leptin, what is leptin? It's a satiety hormone. And so leptin is going to be released from fat cells as fat cells are expanding, and it has part of a sensation of let's withdraw appetite. Appetite has a lot of triggers. There's a lot of triggers in appetite. But when you look at mice, at these leptin deficient mice, in this case, there's a nonsense mutation in the mouse leptin gene. Remind me what a nonsense mutation, Missense Snip? Yeah, the stop codon shows up early. We had a premature stop codon. When you get a premature stop codon, here's the normal mouse. Here's the one with the premature stop codon. Oh man, that thing can't get enough to eat. Same thing over here. The normal mouse and its increasingly galactic brother. And again, there are multiple. There are frameshift mutations. There are missense snips. There are multiple. The first one identified and seemingly the most common is that nonsense mutation in the leptin gene, that premature stop codon. Same thing over here. That's leptin. It's not like you kind of can't blame it, right? Genetically, it just cannot stop eating, and people can get this too. These journals will be available later today in the file folder. But there's a bunch of different leptin mutations in people too. They're not that common. The Belgian blue cow, they're dogs. This is from the whippets, the bully whippet dogs. Here we describe a new mutation in a whippet dog breed, and this is myostatin, right? Myostatin, remember, it's a myokine, a small signaling protein released from muscle, and one of the things it does is stop PKB. So you're describing a new mutation in the whippet dog that results in double muscled phenotype. This picture is from the article. It looks really cute, probably from where you're sitting, because the screen is a little blurry, but it has a striated chest. It's just Ronnie Coleman would be jealous. I mean, just ripped shoulders and a straight chest. And so this double muscled phenotype known as a bully whippet. Individuals with this phenotype carry 2 copies of a 2 base pair deletion on the 3rd exon of the myostatin gene, leading to a premature stop codon at number 313. So if you get a premature stop codon in the myostatin gene, you do not get functional myostatin. If you don't have functional myostatin, you start to look like this. And let's watch a video of a cow showing off its glutes. Look at those things.
That's all muscle?
That thing is ripped. It doesn't enjoy being that ripped. There's no way it's happy about it. Look at that set of glutes.
So what would that taste like?
You know what I love about that question is on the last slide with the dogs. What would that taste like? Well, it wasn't asked about the bully whippets, but with a cow, definitely what would it taste like? And I wonder too. I have the same question. But I do think it's just a tiny bit funny that you show the dog and then you show the cow. And I wonder what that one tastes like. Because I have the exact same reaction. My hunch is, a steak knife isn't enough. You need lasers to cut through it, probably. Like a Jedi sword. Like that's what you need to cut through that meat problem. So I have no idea, but it's selective breeding. When they talk about myostatin knockout, it's selective breeding practices to get them to, that thing. But so yeah, the Belgian blue myostatin sequence contains an 11 nucleotide deletion on the third exon, which causes a frame shift that eliminates virtually all of the mature active region of the molecule. So a frame shift mutation, dysfunctional myostatin, meat that requires a Jedi sword to cut. We'll do a few more of these. You don't write them down. But sickle cell anemia, here we have a little baby Voldemort at the end of, I guess it's the 7th movie or something. Was it 8th?
Yeah.
Okay. Oh, it's 7th part 2. Okay, so a little baby Voldemort. Now, the reason I have a baby there is in adults, hemoglobin, we have alpha chains and we have beta chains. Adults in hemoglobin. And remember, hemoglobin, that's the oxygen carrying component of the blood. And we have 250, maybe 270 million of those things in there. But an adult has a pair of alphas and a pair of betas. Little baby Voldemort there has a pair of alphas and a pair of gamma globins. Those gamma, because you have to be a parasite, right? You have to have a greater affinity for oxygen, that strength of that relationship with oxygen than grown-up incubating mom, right? Because you need a supply of oxygen, and so those gamma globins work better. But shortly after that happens, we replace our gammas with betas. And so we have, everyone in this room, your hemoglobin has a pair of beta chains. In that beta chain, there's a single nucleotide substitution, right? A to T, so it goes from GAG to GTG. And valine gets inserted instead of glutamic acid, and then they sickle, right? The shape is going to be different. We get this sickling, and we can check for this stuff. There are lineages that express it more. So it's great as a combatant for malaria. I mean, great. It contributes to a resistance to malaria, but it's not helpful as a soccer player. And so that's what sickle cell anemia is, those beta globins. Duchenne muscular dystrophy, most common kind of muscular dystrophy. Here's dystrophin right here. And you know, in a muscle, in a skeletal muscle, you're going to have lots of linkage proteins, lots of proteins holding things in place. It's not just actin and myosin doing their business and floating unsupported by corresponding proteins. So you have a lot of linkage proteins. And dystrophin is going to be one of them. There are 9 types of this one. It takes 16 hours to transcribe. Apparently, this is a long one. I mean, just think like Titan, over 30,000 amino acids long. Now, translation can happen a little faster. 500 amino acids, you can get that in a minute. But transcription be a little slower. So, but in Duchenne muscular dystrophy patients, absence of dystrophin is caused by a frameshift mutation in the gene. So you get a premature stop codon and you don't end up getting it. Prader-Willi syndrome. There's a friend of mine in Oregon who has worked with Prader-Willi patients. She's a neurophysical therapist, does in-home physical therapy, and maybe I'll see if she can do a lecture for in-home physical therapy. It gets a little extreme. These are usually the patients who can't leave the house. They can't actually go to the clinic. You have to go to their houses. And so you get some more extreme ones. But she's worked with a lot of Prader-Willi patients. And you just get this huge deletion, right? You get a big genetic deletion for Prader-Willi syndrome. And obesity follows. Marfan syndrome. Now, I don't think Manute Bol, who was on the first slide, the guy who's like 10 feet tall, no, he's no longer alive, but he was super tall. I don't think he had Marfan syndrome, to my knowledge. He didn't have Marfan syndrome. He sure looked like it. And I know people who have Marfan syndrome, and it's something that is now tested for. So this was a college athlete who had Marfan syndrome. And it was just days apparently before the draft. He's from Baylor. They said I wouldn't be able to play basketball anymore at a competitive level. He told ESPN, only back tears. They found the gene in my blood sample. They told me that my arteries in my heart are enlarged. It goes on and on. So Marfan syndrome, fibrillin is the fibroblasts. This is one of the things they're secreting, fibrillin, into the extracellular matrix. That's what the ECM is. And elastic fibers, this is critical to have these elastic fibers. And when we look at, there's a ton of mutations that can cause this. To date, over 600 mutations have been published, but only a minority are recurring mutations. And missense mutations are being found. That's the most common one to see these missense mutations. Now, this article goes over a ton of, this one will be in the folder later, but it goes over a lot of genetic predisposition for pathological conditions. For developmental syndromes and diabetes and cardiovascular disease and obesity and neurological disorders and autoimmunity and cancer. It just goes on. And cardiovascular disease, it breaks it down. There are subheadings for genetic susceptibility, but it's going to be all of these genetic phenomena you already know. Diabetes is going to go break down the data, autoimmunity and inflammatory conditions, cancer, breaking down those conditions. But the major points to take with you are that we have a genotype, we have genes, we have DNA. It lives in our chromosomes. We have 23 pairs of these chromosomes. Our DNA lives inside of those. That's our genotype. Our phenotype is the meat that those instructions, those recipes create. And the nucleus keeps the DNA, right? It sends copies to the ribosome. If you're an architect, let's say, and you're going to go to, you're building Nostalgia 5 in Brookside, and you have 2 floor plans because it's the same thing, but there's like one of them is mirrored. You're not going to go give the guy with the hammer or the lady with the hammer or whatever your original copy. You're going to make copies of it and send those to the neighborhood where the work gets done. You're going to make copies. So that's the RNA. That's copies of the architectural plan. The architectural plan is your genotype. You make RNA, you make some copies of this thing, and send it to the ribosome, and that's where you erect your house. And you're going to have some alleles in there. So your house is going to be unique and uniquely lived in. Later, that's also environmental stresses are going to change your gene expression. Live in that house for a while, and you make it home. Hang stuff on the wall, break stuff on accident, whatever it is one does to a house. Phenotype, right? You're going to change what was your original architectural plan. And you know what that looks like for cell signaling, how things interact and affect cellular responses, how behaviors, how exercise, different characteristics of exercise, how diet, how supplements, how sleep and alcohol, and all of these things, how they affect cellular behavior. So this is, Garrett Ash, we did our PhDs together. This is a diagram I did for one of his articles. And he was looking at blood pressure. And so the use of genomics in the prescription of exercise for hypertension. Somebody has hypertension. Genomic data, as it currently existed in 2013, how do we prescribe exercise to the best of our knowledge? And these are the chromosomes, X and Y and the 22, and these were all the blood pressure related genes, right? It's throughout. It's not, again, it's not like, oh, there's my blood pressure gene. It's a constellation. And, from our chromosomes, our stored, DNA, we get transcription, we get the architectural plans, we get a copy of it, we send it to the ribosome for translation, make some proteins, those proteins fold, interact as biological networks, and then we wind up with a clinical translation. It's a young field, though. Exercise genomics is a young field, and we'll talk about that and age, the effect of deterioration with age, senescence, what we're looking at this age-related kind of atrophy and risk. We'll look at that next time. But what do proteins do? A lot. Yeah, name a few things. Name a few things that proteins do. Enzymes.
I said hormone.
Hormones.
Intracellular functions.
Intracellular, yeah, pretty much everything. Let's just say everything right now, and let's say class. 0.03. Where do proteins come from?
Amino acids.
Yeah, link together amino acids. Where?
Ribosome.
At the ribosome. Link together a bunch of amino acids at the ribosome with the help of those instructions, with the RNA. And again, the transfer RNA, but the mRNA, the messenger RNA, that's the ingredients. tRNA is to get the amino acid. How many protein coding genes do you have? 20-something thousand. Yeah, 20-something thousand. What are codons? Series of 3, a little nucleotide triplet. What's an allele? Yeah, these variants, these versions of genes, slightly different versions of the same genes. What's a missense snip?
Moving shit around.
Yeah, but for frameshift mutation is more like moving tons of shit around. Moving a tiny dump around. Like, yeah, moving around like an almond-sized dump. Moving huge, like shitloads would be the frameshift mutation. What's a nonsense snip? Yeah, premature stop codon. Genotype versus phenotype? Good. Transcription versus translation? All right, you're good. You're good with genes. That's it for the fundamentals of genetics. And again, next time we'll do exercise, genomics, and age. And then I'm hoping that we can get Travis to take the screen as a surrogate for Take the Stage. And we'll follow up, because he's going to be talking about neurology. And so then we're going to follow that up with sort of a step-by-step unpacking of neurology, BDNF, exercise for cognitive health, stuff like that. Pardon?
Is it early day today?
It's an early day today, yeah. Before I have any micro sleeps.